Hello, listeners, and welcome to this episode of Cell and Gene, the podcast. I'm your host, Erin Harris, and I'm so happy to have back on the podcast Dr. Robert Ang. He's the president and CEO of Vor Bio. Robert was one of my first guests back when we launched the podcast, and with all the progress Vor has made since our last chat, I thought this was a perfect time to welcome him back. So let's get into it. I don't get to say this often, but welcome back, Robert, to Cell and Gene, the podcast.
So fantastic to see you again and to be on your podcast, Erin.
Good. All right. Well, thank you for being here, like I said, and let's jump into it. So now, over 2 years ago, like I said, you were one of my first guests. I think so. For listeners, go back and listen to episode 13, and you'll hear our original discussion, which might help even lay some groundwork for what we're about to talk through here. But when we talked about Vor's groundbreaking approach for treating AML, and at the time, Vor had not begun clinical trials to test the approach. And now Vor has treated 8 patients demonstrating clinical proof of concept and are also in the clinic with a CAR T that could be used in combination with Vor's shielded stem cell transplants. And so we have a lot to get through, and there are a lot of really good updates. But before you do share those updates with us, Robert, talk us through kind of broadly about AML, how far the industry has come, and broadly speaking, what does the AML patient journey look like?
Sure. So a lot has changed in 2 years, but I'll say AML is one thing that hasn't changed, Erin. So for those listeners who are less familiar, AML is acute myeloid leukemia. It is known as a blood cancer, but actually it's a cancer that centers around bone marrow, because these cells, these myeloid cells that become malignant, are cells that typically reside in bone marrow. And one of the difficulties with these cancers is that, firstly, they replicate uncontrollably like a lot of other cancers, but because the bone marrow niche is very is compact in size, they tend to start crowding out other healthy cells within that bone marrow. And so it almost squeezes the life out of you. So the bone marrow reconstitutes all the cells necessary for your circulation, which includes all your red blood cells, all your immune cells, platelets. Those cells start decreasing in number, called cytopenias, and the cancer kind of bleeds you dry. And the other issue is that AML, since it's in the bone marrow, it almost behaves a little bit like a solid tumor in that way. So it hides out in these niches. One of the strategies that was pioneered literally 50 years ago now is that you could use high-strength chemotherapy, other toxins, to eliminate all the cells in your bone marrow, of course, all your cancer cells, but it also eliminates your healthy cells. But the discovery 50 years ago is that you could actually transplant in cells from someone else, what's called an allogeneic transplant, and have these cells reconstitute a new blood and bone marrow system in the patient. And I mean, totally revolutionary that you could actually take foreign cells and have them grow inside a patient and essentially reconstitute their entire blood system for the rest of their lives. And that meant you could treat some of these cancers that were otherwise totally resistant to other forms of treatment, including chemo. Unfortunately, we are still in a circumstance where half the patients who get that very traumatic stem cell transplant, they also relapse their cancer. So we gotta improve on that, right? We've got to do something more for these patients that gives them hope that they can foresee a permanent cure down the line that is worth all this trouble that they're going through.
Sure. Okay, good. Thank you for level setting that conversation for us. And the more, which you're referring to, we'll get to in a little bit. And before we do, I hear a lot about, and I'm sure our listeners who are familiar with Vor hear a lot about the Vor approach. And I'd like to talk through a little bit about exactly what is the approach and what are the steps to it.
Sure. So Vor is taking quite an orthogonal angle to treat AML. Let's talk about some of these historic efforts to treat, cancer in general as well as AML. We know chemotherapy. Chemotherapy poisons, that they poison cells that are rapidly growing or, a particular phenotype that happen to be cancer cells, but also happen to be other cells. And so you get a lot of attrition, right? You get a lot of toxicity because you're killing cancer cells as well as healthy cells. That could be as simple as your hair falling out or, getting, bad diarrhea, but it could also definitely put the patient's life at risk. And I called it a hammer. So, you're basically hammering whatever looks like a nail. Some things aren't nails, but you know what? Too bad. We've tried that in cancer. We've tried that in AML. Unfortunately, in AML in particular, it's incredibly toxic because you start attacking your own blood system. These patients end up getting these cytopenias with low blood counts of cells, including white cell counts, which you cannot replace. And so they're subject to all kinds of infections and issues that sometimes they cannot recover from. So the orthogonal approach that Vor is undergoing is, what if you didn't develop that another hammer? We've tried the hammer approach time and time again. What if you instead actually deliberately hit the healthy cells? And so the only thing that looks like a nail is the cancer cell. And so the healthy cells are simply preserved and functioning and very healthy in the face of this big hammer. And so to do this, we are hiding healthy cells by genome engineering the patient's stem cell transplant to delete out certain proteins or certain epitopes that allow these cells to be invisible. We call it shielded. So you're shielding these healthy cells, but you're still exposing the cancer cells. And we're doing this initially with an approach involving a protein called CD33. Now, back when we last met, we were still not in the clinic. We were relying on animal data and also genomic data, which described that CD33 we don't know exactly what it does, but we don't think it's super critical. And in fact, we found out that basically 1 in 10,000 people, healthy people, do not express CD33. And that's kind of an interesting factoid, right? That occasionally you come across these people that just don't express CD33. So they seem to be perfectly healthy. Maybe you don't need this protein. That is a really interesting observation, but it's a different question to say if you or I, having expressed CD33 our entire lives, if we start receiving a stem cell transplant that lacks CD33, what happens? Do these cells behave normally? Do they behave with all the other cells that do express CD33? Are these cells selectively growing or diminishing? Do they act like a regular blood system? And that's a question that we could only answer by entering the clinic and treating patients.
Good. Okay. Thank you. Well, speaking of entering into the clinic and treating patients, tell us about all the progress that's been made since we last met, including progress with VCAR33 Allo.
Yeah, sure. So we're privileged to have 2 programs in the clinic. One you're referring to in the CAR-T, which I'll get to in a second, called VCAR33 Allo, but Trem-cel, as it's now known, used to be called Vor33, but Trem-cel is our shielded stem cell transplant that I referred to. And so what we're doing with this drug is we're taking a regular stem cell transplant, but we are genome engineering it to miss CD33 expression. So now you've got cells that hopefully behave like regular cells, stem cells, but they no longer express this one protein. And we've managed to read out 8 patients worth of data to date, showing that 8 out of 8 of these patients managed to reconstitute their blood system and have what's called neutrophil engraftment, meaning that the stem cell therapy can actually reconstitute neutrophils, which are very important white blood cells that protect against infection. And they can also reconstitute platelets, which obviously help your blood clot. 2 very critical cell types that you really, scrutinize very closely after a transplant. And so that was fantastic news for us because, again, before entering the clinic, we didn't know whether that was possible. The other thing we're doing in that trial, the Trem-cel trial, is we are dosing patients with Mylotarg. Mylotarg is an antibody drug conjugate that detects CD33. Mylotarg is very famous for being incredibly toxic. So again, everything looks like a nail with Mylotarg, and so it attacks a lot of these healthy cells, which then give you all these cytopenias. So it essentially wipes out your blood system. In 3 patients that we've disclosed so far who went on to receive Mylotarg, their blood systems was essentially fully intact. That we could dose Mylotarg in a way that has not been done before without the dramatic cytopenias you typically see within days of Mylotarg treatment. And in fact, patients could receive multiple doses without any of that cytopenia occurring. So that was really revolutionary data of its time, and we're continuing to enroll patients and increase Mylotarg dose and demonstrate that important proven concept. Now, you asked about a second program, which is VCAR33. VCAR33 is our hammer. We need to couple Trem-cel, our shielded transplant, with the big hammer, which is VCAR33. And that is a program we're super excited about. It entered the clinic and we treated our first patient just in January. The clinical progress is going super well there. We're expecting to release data later this year. And VCAR33 is unique as a CAR T because unlike any other CAR T that has hit the clinic with a commercial manufacturer, we are not using cells from the patient, which is called autologous. Neither are we using allogeneic off the shelf, which other people have done. Both approaches are somewhat compromised. Cells from the patient, autologous, these patients have gone through a journey we've described, including some very traumatic treatments, and this T cells may not necessarily be in their best shape. Allogeneic off the shelf approaches have somewhat disappointed in the clinic, despite multiple different approaches, and it's really simply because it's hard to hide these cells from our blood and immune system, and they tend to get wiped out quickly, and I don't think really get the best chance at showing efficacy. What we're doing is different. It's neither of those things. We're using cells derived from the prior transplant donor. And so we are taking cells that are healthy. Certainly the donor has never seen chemotherapy. But on top of that, this transplant donor previously reconstituted the blood and immune system of this patient. And therefore, these T cells that we're harvesting are exactly matched to the patient, while still not being technically from the patient. So we're super excited at this. These are really healthy cells. We've started dosing, and we're really looking forward to that next data disclosure.
That's incredible. And certainly for VCAR33 Allo, we'll have to have you back again to discuss that data once it becomes available.
Well, I'd love to.
Good. Okay, so thanks for that, all of that detailed explanation of what's currently going on. But I want to talk now a little bit about, so back in September of 2022, Vor opened a new in-house clinical manufacturing facility in Cambridge. And anytime a company invests in their own in-house manufacturing facility, I'm always interested in knowing the why and the how. And so, why the investment in your own internal cell therapy manufacturing facility? Why was co-location with your headquarters so important? And what's all being manufactured there? Yeah.
So, Erin, you've come across many companies making their own facilities. It's done for, certainly some similar reasons, but some different. Our philosophy of Vor is that we really want to own and control our own manufacturing process. This is really because these cells that we're manipulating are going into the patient, and they are living drugs that are doing their work over months to years, and they need to be as healthy as possible. And so our vision with our internal manufacturing facility is that we want something where we can go straight from a process development or research into process development environment, and then straight into a manufacturing facility that we could literally, do it across the whole way. And that is exactly what we set up at Vor. And so we've enabled VCAR33 manufacturing in-house. In fact, that's, our operators are there making clinical product right now for patients in need. That allows us, again, to own and control this manufacturing process. At the same time, we're also a believer of using CDMOs to really help us, where we can. And so we've had a CDMO make Trem-cel for us from the get-go. They are still making Trem-cel for us right now. And, we're in the middle of thinking about what does phase 3 look like? What could commercial look like? Again, we're not set up to make our own commercial product here. I think we would have overinvested if we had made a commercial facility. And so, that's not to rule out that we couldn't do it ourselves later on, but we want to make stage-appropriate investments, as we make progress. Now, you asked the question about, why is it important for co-location? Corporate culture is like super important to Vor. And having some folk siphoned off in another part of Massachusetts or wherever it is, or another state, starts making a kind of two-class culture in a way. We are on multiple floors in this building. We even have cultures we know that are different across floors. But at least we're all in the same space. And so for us, this was a luxury that we managed to work out from a very flexible landlord and a good investment in HVAC and other things. But having it here and our manufacturing team and quality teams all fully integrated in the company just makes it so much easier that we can really act as that united one company.
Yeah, and that makes a lot of sense. And I do like how you talked about the hybrid approach of, in-house and CDMO use. I think that's really smart. And I think a lot of our listeners will have learned quite a bit from your experience and what you just detailed. So thank you. All right. Talk us through, what does the rest of 2024 and maybe the foreseeable future look like for Vor?
Yeah. So, what we have are these two programs. So Trem-cel, we have a CAR-T. What's important is that not only do they stand alone as clinical programs, but they also work together. So Trem-cel patients, we're enrolling patients who are at high risk of a relapse after the transplant. We know that just based on molecular markers of disease and other risk factors. And so we know that, unfortunately, a lot of these patients are inevitably going to relapse. However, what we put in place are important treatment options that help to give the patient opportunities if or when they relapse. And that includes enrolling into our CAR-T study. So we deliberately asked FDA if we could enroll Trem-cel patients who may relapse or become MRD positive, and they can roll straight into our CAR-T study, which again now provides this hammer, which we think would be very potent, but to patients who already have an established blood system that is shielded because of the genome engineering we're providing with them with Trem-cel. That is super exciting. And that's what people have been waiting for ever since Vor was conceptualized. And so this year is going to be a really important year for us. I think we'll certainly read out more patients with Trem-cel. We're increasing dosing of Mylotarg, and that's going to be exciting. We're reading standalone patients, in the CAR-T who may have relapsed after a regular transplant. But we also have this possibility that Trem-cel patients can be treated with the CAR-T, and we can really start to see what this combination, what we call a treatment system, really looks like in Vor. So I think it's a super important year for us. This is also where the rubber hits the road, but something we're embracing with open arms. And I think it could be transformative for us and for patients in need.
That's amazing. And we, like I said, Cell and Gene will be following you along your journey. And so we'll have you back and, maybe this time next year to talk about all the progress.
Oh, that'll be really exciting to do. I'll be your first guest to appear 3 times.
That's repeat, right? Yeah. Good. Well, we've come to the formal end of our discussion. And so last time we talked, again, for listeners, that's episode 13. Go check it out. as my listeners hopefully now know, I like to talk to my guests about their interests when they're not developing and manufacturing and commercializing life-saving therapies for the patients who need them. You mentioned that you spend some of your spare time making, I think I'm going to say this right, vacuum tube audio equipment and amplifiers, which is so interesting. And so I'm just wondering, are you still doing this? And if you are, how have you progressed in the last 2.5 years since we talked?
Yes, Erin, I'm still doing it. It's still something that keeps me sane. I happen to be building them and keeping them to my wife's chagrin, and they're taking up room in my basement. But I think as your listeners know, I was a surgeon previously, and this is essentially some of my surgical outlet. I can get my hands dirty, elbows deep in an amplifier chassis, and create these things. And in a way, it's kind of similar to what we're doing in the clinic right now. We are making these little factories, these little machines, which are cells that are doing certain things when we allow the patient to be treated, and that's essentially what these amplifiers are. They're like electronic equipment, but you turn them on, and suddenly they come to life. They have their own voice, and you can actually tweak them in different ways to change that voice. So it's awesome. It's an outlet for me. It happens also to be a bit of a risk factor, since occasionally I do electrocute myself, but don't tell our D&O firm and my life insurance provider. But a ton of fun, and it's great to have an outlet.
That is so cool. And yes, your secret is safe with me and all of our global listeners. We won't tell anyone. It's just I love hearing what my guests do, and this has to be up there with one of the most interesting hobbies, things that people do in with their free time, and to kind of enjoy what they're doing beyond work and what they're doing in the lab. So that's incredibly cool. All right, well, listeners, that wraps up this episode of Cell and Genome Podcast featuring Vor Bios' Dr. Robert Ang. Robert, thank you so much for being here. This is great.
Oh, it's a pleasure, Erin. Thank you for having me.
Absolutely. All right, listeners, thanks for tuning in, and we will talk to you soon.